Okay, awesome. Um, so I'm really excited to welcome our first speaker, Carol. Carol, you're going first? Okay, awesome. She'll be presenting her work on the monophyly of Compositae. So this will be a wonderful talk, and I'll hand it over to you.
All right. I just need to do a little um, shuffling here with my screens to make sure that I'm sharing the right thing. Okay. Let me get started. Okay. All right. So I hope everybody's seeing my screen now. Um, so I'm so happy that I can present this today. So this talk I prepared to present on the Botany Conference this year. So that was in July in Palm Springs here in the US. And I did get some people that is in this meeting to come and watch. So if you were in the meeting and you watched it, you're gonna watch it again. But I'm happy that I can actually talk to people that are directly impacted by some recent results, which is the Asteraceae Synenterology community. So let's get to this. So probably everybody here in this meeting room uh, saw this paper came out last year. This is basically the capstone paper of the first phase of the PAFTOL project. So it was a gigantic effort to summarize the results from that first round of sequencing into a publication that could present all those differences that they found in relation to the current known phylogeny of angiosperms and differences in classifications and things like that. So for us that work with Asteraceae, you probably read the sentence saying that one of the most striking things that they found was that Asteraceae was non-monophyletic in the tree. And then if you go to the tree that they present as the global family level tree, the reason that Asteraceae came as non-monophyletic is that uh, Calyceraceae, which is considered the sister group to Asteraceae, uh, is found nested within Asteraceae, sister to the Barnardesioideae, to the subfamily Barnardesioideae. So if you've never seen a Barnesioide in your life, this is what they look like. This is a dasyphilum. So Barnesioide are very interesting. They are mostly woody plants. Some of them are trees or like very large shrubs that kind of climbable things. They have this pair of um, spines subtending the leaves, so the stipules are modified into spines. And then you have a very firmly imbricate um, involucre with lots of series. And then we have a lot of different of specific morphology of the stamens, of the stigmatic surfaces of the styles, and so forth. So they're a very interesting group, and they have caught the eye of a lot of people for a long time. But this this note in the paper that Asteraceae was no monophyletic in their analysis caused kind of a lot of ripples around the synenterology community and also the plant systematics community in general. So for those of us that were at the IBC last year, this was presented at the IBC. There was a lot of talk about it. Uh, People have been fielding questions about this. So Jennifer Mandel got a lot of questions about this in other occasions. So because of that, we wanted to take a better look at what was going on. So one thing that I want to mention is that I'm not here trying to diss anybody. I'm not gonna like this is not I'm not criticizing, well, I'm criticizing some of the results, but uh, this folks that actually worked on the paper, they are my co-authors on this paper. And I'm very glad that they were so open on me poking around the data and seeing what I found. So I'm very glad for this collaboration and for the opportunity to work with the folks that are doing the PAFTOL analysis and understanding this massive amount of data that they're generating. So once they were analyzing the data, my colleague Benoit, who I don't know if it's here in the meeting or not, he came to me and said that they were very confused because Calliceraceae were falling within Asteraceae. and everybody was kind of like nervous about it. And I was like, well, we've actually seen this before. So in 2019, you guys might be familiar with this paper. When I was working with Jennifer at her lab, we published this first look into the phylogeny of Asteraceae using target capture data. So in this paper, we had a pretty good sampling of almost all the Asteraceae tribes. We were only missing a couple. And we actually had some outgroups in there in the two Goodeniaceae, one Calyceraceae, but we were not super particularly interested in understanding where Calyceraceae and Goodeniaceae were falling or what were they were doing because we were interested in the Zing group of the Asteraceae. So if you look at this 2019 paper, in the tree that is in the main publication, you see that Calyceraceae is where it's supposed to be. It's sister to the rest of Asteraceae. But when we were doing the analysis for that paper, we recovered actually Calyceraceae nested within the rest of the Asteraceae when we are using the Compositae 1061 dataset, which is the main dataset that we present in this paper, um, and doing an astroanalysis. So we see that the only terminal of Calyceraceae that we sampled, that was Nastantas. It fell within the Asteraceae, and it was not even sister to Barnardesioideae as the Paftol paper recovered. It was sister to the rest of Asteraceae after Barnardesioideae. So because we were not super interested in the outgroup question, we only had one species of Calyceraceae sampled, which is important here because this type of spurious relationship can be caused by an imbalance in your sampling. We didn't really discuss it, and it went into the supplemental materials. So if you download the supplemental materials, this is figure 4 there. And also in 2019, there was still some discussion in the literature about using pseudo-coalescent methods like Astral in relation to concatenated analysis. So we ended up presenting the concatenated figure in the paper, which was most accepted in the time that we published this paper. So we were not particularly surprised that Calyceraceae was doing weird things in the Paftol paper. So one thing that we should notice, though, is that to assemble this massive tree that is shown in this 2024 paper, they had to do a very elaborate backboning strategy to run this phylogenetic analysis. So if you ever have to run a phylogenetic analysis with thousands and thousands and thousands of terminals, if you decide to just run gene trees to do like a coalescent analysis or a concatenated tree, you're going to sit and wait forever, basically. So our current phylogenetic methods, maximum likelihood, Bayesian analysis, they are not particularly good in dealing with very large alignments, with lots and lots of terminals, even if the alignments are short. So Bayesian analysis is completely out of the question here. There are no methods that can take in a matrix this size. So we end up relying on the maximum likelihood analysis. And even then, if you were going to run one gene with 12,000 terminals, this analysis is never going to run, or it's going to take years to run. So to avoid this like very long and intensive computational process, that I should mention that these things don't like the only thing that you're not only wasting time when you're doing this very large analysis, you're also wasting computational resources that depend on things like water usage from data centers, electricity usage, and we should be conscious about that in our role as scientists. So Paftol folks did a backboning strategy. So they basically did a subsampling of taxa and they ran several analyses to see if the positions were kind of similar. They used different types of data sets. I'm not going to go too deep into the methods that they use, but it's important to remember that to assemble the backbone of the tree, so the relationships between the families, they were not testing the full data set with all the 15,000 species. They were using a subset of species. So we are reducing the size of the data sets in each pass of the analysis, basically. So my first thought is, so if because of this backboning that they did, we might need to reanalyze the data without applying these constraints, right? Just let's reanalyze the full data set. And that's what I did. And the main result of that analysis is what I would like to say. Could the reports of composite non-monophily been greatly exaggerated? Paraphrasing Mark Twain here. And I would say that in parts, yes, it was a bit exaggerated. So I ran two different analyses. So I asked uh, our PathTalk collaborators to send me the full alignments that they had for the Asterales order. So the whole order. So all the families that they had sampled. And they sent me this data set, and I basically just ran two analyses. I reran the Astro analysis without reducing any number of terminals, and I ran a concatenated analysis on the same data set. And and when we look at the tree that was, and I sorry, I kind of like skipped over what I did. When we look at the tree that was published in the paper in 2024, if you go deep into the extended figures, you see that this relationship between Barnardizioides and Caliceraceae has low support. So the support here is less than 0.5%, 0.5. So we would consider that this is like basically a collapse clade if we were taking the support values into consideration. So in parts, this exaggeration comes from basing these claims on low support. And then once we reanalyze, sorry, I said everything that I should have said in this slide before. So reanalyzing the data set, both of the astral and the concatenated maximum likelihood analysis, we recover the traditional topology with Caliceraceae sister to Asteraceae in total, not just Barnardizioides. So, and this has very high support. It's like one, like full support here, full support here. We get very good resolution within Caliceraceae, within Barnardizioides, more or less Barnardizioides, but like we don't see any like obvious problems that would point out that like there might be some sort of incongruence going on in the backbone here. Because we have full support, nothing like the both methods recover the same topology. So I wanted to dig a little deeper in the data to see what was going on actually. So, and there are multiple ways of doing that. So I wanted to understand this incongruence signal that we were seeing and how we can better define it. So we know that sometimes, like everybody uses alignment statistics to understand if an alignment is good enough to be used in phylogenetic analysis or not. So there are some things that we could look into, like the length of the alignments, the number of parsimony formative sites, the number of taxons. I could look straight at the gene trees and see one by one to see what these tree groups are doing in these gene trees, but like this is a lot of work and it becomes confusing very quickly. We could look at 5 parts or quartet sampling that are like these traditional ways of measuring congruence in across gene trees. But to me, these were not exactly what I wanted. I wanted to understand what the alignments were doing, not what the gene trees are doing. So I was trying to figure out what to do when my colleague Benoit pointed out this paper here to me. So I would never have found this paper on my own because uh, it's not even plant phylogenomics. So I would never go out and look for a paper in such a different area than mine. But Benoit pointed me to this paper, and I looked at it. I was like, "Oh, this is a really cool method." And it is a kind of a similar question to what we are trying to do here. And this paper, so the paper is from 2021, so from several years ago, and they use a method to look into this differences in topologies that I thought it was very interesting. And it was based on this method called likelihood mapping, which comes from this paper from 1997. So it's a method that has been around for a little while, but like I really have never seen people using it. So I decided to go ahead and try to get this done with our Asteraceae problem. So because of computational time and real life time, I decided to reduce the data set a little bit, but in a more intentional way, not just trying to like pick something here, and something from there. I wanted to make sure that we keep the groups that we are seeing this issue with intact. So I trimmed that Astralis tree that has about 2,000 tips. I trimmed it to 46 terminals. So I got all the Miniantaceae, I got all the Goodeniaceae. So I'm keeping the Miniantaceae, the Goodeniaceae, well, the Miniantaceae specifically as the outgroup here. And then the Goodeniaceae was kept intact. I got all the sampling of Calyceraceae that they had. I got all the sampling of Barnardesioideae that they had. And I reduced the core Asteraceae to 21 terminals. So if you see, I kind of skipped over some of the South American groups like Mutisioideae and some Symphyotrichum, Wunderlichioideae, and I went straight into the core Asteraceae, which basically starts in the Cardomoideae. So I reduced that, and I used this tree as my basic tree. So with this reduced tree, I then I manually created 3 different topologies. So one is the traditional topology where Calyceraceae is sister to all the Asteraceae. The second is the topology that we found in the Mendel 2019 paper, where Calyceraceae is sister to Asteraceae with the exception of Barnardesioideae. So Barnardesioideae then is sister to Calyceraceae and the rest of Asteraceae. And then topology 3 is the topology found by Zuntini et al., where Calyceraceae and Barnardesioideae are sister to each other, and this group is sister to the rest of Asteraceae. So with these 3 topologies in hand, then I trained all my alignments to match the sampling. So all my alignments then had 46 terminals. And then I used IQ3, which is a maximum likelihood analysis program, to calculate the log likelihood of each alignment fitting with each constraint. So basically, for each alignment in my data set, I ran it 3 times in IQ3 to get a likelihood of that alignment matching each topology. So in each one of those runs, a different tree was the fixed topology that the alignment need to match, basically. So in the end, for each alignment, I have 3 measures of likelihood, 1 for each topology. And you see that this vary a lot within each alignment. So sometimes the numbers are very similar, sometimes they are very disparate across the data set. So because the likelihood values numerically vary a lot, I then normalized the likelihood values so they would range from 0 to 1. So I could fit all of them in the same universe. And then I basically chose the best topology for each alignment. So in this process, basically, I would get the highest value, the better likelihood for each alignment, and I would select that as the chosen topology. So for some of them, for example, this 2 here, this is clearly one of the 3 topologies. So in this case, it would be topology 1. And for some of them, the values are so similar among each other that then we have an incongruent alignment, not an incongruent, an inconclusive alignment that could fit either one of the topologies, either the 3 topologies or 2 of the topologies equally. So after this was done, I then used a very fancy way of representing this data that is called a triangle plot. So let's walk through this slowly. So each one of these dots represent an alignment in my world of alignment. So I have, so this is the Angiosperms 353 data set. So we have about 353 alignments in here. So the position of the dots within this triangle indicates the topology that alignment most closely aligns with. So the closer to the tips of the triangle, the more strongly it fits with that topology. So for example, this group here fits very strongly with topology 1. And then the farther away it gets, the less it agrees with any one of the topologies. So in this darker triangle here in the middle, these are those alignments that are considered inconclusive. So the ones that don't really strongly match any of the three topologies. And then on the sides, we have the alignments that kind of match a pair of topologies. So for example, an alignment here could match either topology 1 or 2 more or less equally. So it's hard to say which one it is. And at the same time, I counted how many alignments fit within each one of these categories. So what we see here is that approximately almost 100 of our alignments, they fit better with topology 1, which is the classic topology with Calyceraceae sister to the rest of Asteraceae. So you're like, okay, then that means that we don't have any issues. We can just like believe that this is the right phylogeny and we don't need to think about things not being monophyletic or not. But another thing that I want to point out here is that almost the same number of alignments, almost 100 alignments, like I think it's 96 is the precise number, they also are inconclusive. So they don't match any of the three topologies more strongly than each other. So this would be non-informative, uninformative alignment. So they either are very short or they don't have enough variable characters. They're those that tend to not do a lot of good in the phylogenetic analysis because they don't have enough information on them. And then we have a good chunk of alignments, 60 alignments almost, fitting topology 2, and a little smaller chunk fitting topology 3. So one thing that I want to point out is um, there are alignments that actually do not fit with the traditional topology, but the topology that we found in 2019 with Calyceraceae sister to the rest of Asteraceae is actually more common than the topology that was found with the PAFTOL dataset in that backboning strategy. So there were, I think, unlucky things happened during the backboning strategy that ended up leading to this topology appearing as the topology that was chosen as the topology for that paper. And then we have a good, like a smaller chunk of alignments that are more or less inconclusive. So they navigate, they fluctuate between one or two between two different topologies. So, okay, what can we do then? I tried to see if there was any differences in the alignment length that could lead to this differences in topology. And you see here that I plotted the alignments that fit into each one of the topologies. I plotted their distributions, and I put this blue line here to represent the median value for topology 1. And you see that like for topology 1 and topology 3, the median value is actually very similar. But there is a difference between the topology 1 and topology 1, 2, 3 that is that is statistically significant based on a Tukey honestly significantly different pairwise comparison test. So we could see that, for example, we could say that if we remove those short alignments, and this is what is represented here, these are short alignments. If we remove some of the short alignments, we might create more certainty towards either one of the topologies. But also, there's not really, well, there is some difference in here. So you see that topology 2 tends to have shorter alignments within the topologies that were precise, but these are not really significant. So alignment length it could be a representation of something that is uninformative, but also cleaning up the data set would not result in things being more skewed towards one or another topology, probably. I then went to look at parsimony formative sites, and I found something similar. There is not a lot of difference. One thing that is interesting, though, is that for the uninformative alignments, they can either have too few or too much variable characters. So it's interesting to see that. So it's not as intuitive as we could think that like the more variation, the better to resolve a phylogenetic tree, but sometimes too much variation doesn't resolve a phylogenetic tree either. And here we see that there is a difference between topology 1 and 2, the alignments that fit topology 1 and 2, and the alignments that fit topology 1, 2, and 3, and 1. Okay, so I've done all this, and what do I conclude? So what we know now is that for the Angiosperms 353 target capture data set, um, for Asteraceae, about a third of those genes were uninformative in these focal relationships. Of course, they could be more informative towards the other parts of the tree, but in this particular problem that we had here, they were not. So we see that there is a real signal of conflict between these alternative topologies. So we have real alignments that prefer one or other topology. We have to remember this Angiosperm 353 is a fairly conservative set of genes, so that should be included somewhere in our thought processing there. I didn't pay attention to any other evolutionary process that could influence in this conflict here. So I don't care about gene duplications. I didn't care about hybridization. I was just looking at the alignments. I don't know. I didn't do any specific treatment to potential paralog genes. I just grabbed the alignments that we were using in the Paftol paper, for example. And I had to reduce the alignments, right? So we don't know if this pattern would hold on with the complete alignments or if it would change, for example. And I also don't know if this pattern holds with a different set of genes. So if I was using the composite 1061 data set here, I don't know if these results would be similar. So this is a very like this analysis is very biased towards the Paftol data set because that was the question that I had in my mind. One thing that I want to point out, though, and I'm almost done here, is that in these uninformative gene trees, cheliceraceae sometimes emerge close or within the mutizioidy. So here's cheliceraceae, and there's a little weirdo here so you can see what they look like. And you see that this cheliceraceae are falling right within the mutizioidy. And you see here I have a parasia, and here I have another weird mutizioidy here. So and I saw this multiple times across the gene trees. So it's something interesting that we can probably think about. And one thing that I want to point out about it is that these groups have a very long evolutionary history in South America during the same period. So Caliceraceae, Barnardizioides, Mutizioides, they were all sitting together in South America for millions and millions of years before the family kind of exploded and moved out of South America and exploded in other places of the world. So if we look at the current distributions, and these are maps that I took from iNaturalist, so take it with a grain of salt that they might not represent the full distribution. So Caliceraceae and Barnardizioides still overlap a lot in their distribution, and the Mutizioides still overlaps a lot in their distribution. So we need to understand this Caliceraceae better. I think we were not going to be able to progress in our understanding of the early history of Asteraceae until we understand Caliceraceae better, and until we understand the genomic evolution processes that occur in these lineages that stayed in South America for so long. One thing that I want to bring in is this paper that got accepted recently. Is still in press. It's going to come up on applications of plant sciences next year. So in this paper, we looked very deeply into potential hybridization events in the Asteraceae history, and we tested it in various ways using the composite 1061 dataset. And we found the involvement of Caliceraceae and Barnardizioides in some events. So we didn't directly tested Mutizi with Caliceraceae, which is something that now I think we can potentially test in future papers. But we see that there is some involvement in potential events of ancient hybridization between different parts of in this part of the tree that has stayed in South America for a long time. And here's a different analysis in the same paper that you see that there's some involvement between Goodeniaceae and Barnardizioides, which is something that we didn't look at before. So Some suggestions for the future based on all that is that we need more genomes, not only in Asteraceae, but in the sister groups as well, because this will be the most direct and best way to understand the hybridization hypothesis that we have. For example, could Bondesioides be a hybrid lineage? It could, but right now we have no way of knowing it. We need a better phylogenetic sampling in Calyceraceae and Guderiaceae, especially with data sets that we can then integrate with Asteraceae so we can understand it, like understand this problem better. And I think we need to embrace phylogenetic uncertainty. So we might not ever know what exactly happened in that split between Calyceraceae and Asteraceae and Barnardizioides, because we might just not have enough information. We might not have enough extant lineages to test that in. And I think that should be embraced by the community and tested in many, many ways. Okay, so I want to watch you all for watching my talk today. I want to thank again the PAFTAL project and the PAFTAL team for the gracious um, uh, this making the graciously making this data available to me. The University of Memphis High Performance Computing Center, the computer resources of Dr. Ryan Folks and his lab, and everybody that makes their data and their scripts open access. I really appreciate y'all. And this is one of my current passions. So if you ever want to make your stuff accessible, please talk to me. And that's it from me. And we can have questions, I think. I don't know. Lizzie, you know if we can have questions or not.
We can. That was really awesome,
Okay.
Carol. Thank you so
No
much.
problem.
I just want to, I think we have a few minutes for questions, but we'll open, it up at the end, if there's more, but I just want to leave time for our second speaker. I see that Alexandra had a question. Do you feel comfortable asking in your own words, or would you like me to read that out loud?
Oh, okay.
More worried if you can actually hear me.
We can.
Yes, we can.
Okay, good, good. I had some sound issues earlier. Thank you. Sorry, so my original question was, I wasn't quite sure if I understood correctly with your fixed topologies, if you fixed the entire topology or just the crucial nodes and let the rest float free. But I also wonder now, towards the end, so starting with a comment. So my understanding of the original PAFTOL paper was that they did not take paralogy into account at all during their bioinformatic assembly. That they kind of did a shortcut there when they based their PAF tools on what was originally HiPiper, that they basically didn't deal with the paralogy recovery that HiPiper originally had. And so I'm just kind of wondering here, to what degree do you think that might be the issue? And also with the test that you did then on, these inferred reticulations, did that test explicitly exclude the possibility that it's deparalogy that's causing the conflicts? Thanks.
Okay. So I only fixed the three nodes. I didn't fix the so I fixed the three nodes. The topology within the clades is the same in the three fixed topologies that are produced, but the options that I used on IQ3, they fix the whole topology. So it's the fit to that topology specifically as a whole. I hope that made sense. And the the PAFTAL paper, yes, you're right. They did a shortcut. Um, Looking at the number of paralogs that are potentially assembled for this part of the tree, I would not be especially concerned with it because it's actually a handful of genes that have some sign of paralogy, at least as recovered in Hyde Piper in that part of the tree. The Angiosperm 353 dataset is actually, you could say, very good with Asteraceae if you don't want paralogs. It is very good in this sense. There's only a few tribes that actually have a lot of paralogs on Angel 353, and they're like Nafali, Senescioni, which of course they are. We expect that these are going to be the problem children. But yeah, but And I completely forgot the third part of the question. I'm sorry, Alexander, if you can repeat it.
Oh, that was more whether your phylonet analysis would exclude that. But if there's very little paralogy in that part of the tree, that's perhaps not in it. Oh no. Well, your analysis was broader. I don't want to monopolize this, but I'm just kind of wondering on the effect if you fix the entire topology, how much of the, mismatch between an alignment and the topology is being driven by the vast majority of nodes that you actually don't care about. But yeah, I
Yeah,
guess I'll have to think about it. Yeah, thank you.
yeah, I'll have to think about it as well. Yes, and I share this concern too a little bit.
Do you want to
Yes,
ask
Nicola.
a short question? And then anything else we'll leave till the end for a more broader discussion.
Sorry, was that me that you were
Yeah.
inviting? Yeah. Okay. Um, yeah, thanks, Carolina, for a great talk. I've got two very quick questions. The one is following on from Alexander, which is okay, so you weren't looking so much at the gene trees, but I was wondering whether within those clades, your 21 Asteraceae, the Barnardizioideae,
Hmm.
Cheliceraceae, whether the topologies within those clades changed much. Um, but I suppose
So
that's not something you investigated.
I actually did because I actually looked at all the gene trees individually. Yes, it changes a bit. Yeah.
Okay.
And in some cases, and I bet these are the middle, like the uniformative gene trees. Sometimes we have like part of the Barnardizioideae in one part of the tree and part in a different one. Sometimes they're in a grade. So there's a lot of variation in the gene trees.
Okay. Okay. And then I was just wondering if you ever did exclude those T123 genes, those 96 uninformative genes, and see what the result was.
I was going to, I thought about doing this before this talk, but I didn't because I didn't have time. But I think
Okay.
I'll do. So we're writing this up to publish a comment on the PAFTAL paper, and yeah, I think I'll do that before we put this in a paper format. Yeah.
Cool. Looking
Mhm.
forward to it.
Yeah, thank you.
Thanks so much for the nice talk. Let's move on to our second speaker, and I'm happy to welcome Sushil Dahal, a PhD student, also based at Mississippi State, and they'll be presenting their work on Symphyotrichum, for the talk today.
Thank you. Give me a second. I need to fix this. Oh. One more second. Where are you? Okay. For some reason, my PowerPoint keeps kicked out.
Huh. Are you sharing the right screen?
Uh, okay. I am. Okay. I think I can now. Hmm.
Do you want to send the PowerPoint to me and I can share it from here?
Yes. Yes,
Okay.
I'll do that. Hmm. Okay, I just WhatsApped you.
Okay.
Yeah,
Uh, yeah.
that's the only
Let
thing I
me.
can open at the moment.
Okay. I'm opening it. Just a second. Okay, and then you just tell me next when you
Oh, okay.
when you need me to.
Oh, actually, I
Is
can't
it
see it
working now?
now. Yeah,
Okay, great.
yeah. Thank
There you
you.
go.
Sorry. Okay. Now everybody can see my screen, right?
It looks awesome, but we can see your
Oh yeah, okay.
the
Sorry.
like
Yeah, you don't want to see all
editor
that.
view.
Yeah. Okay. There you go.
That's
Okay.
perfect.
Okay.
Thanks.
Sorry, everybody. It took. Okay. My name is Sushil Dahal. I'm a recent PhD candidate at the Folk Lab where Carol did her postdoc, and I work on this genus called Symphyotrichum, which is a mest. And then I published few papers, but this one is one of my chapters from my dissertation where I investigated the habitat shift in subgenus Virgulus, utilizing three different approaches: morphological, ecological, niche, and then the phylogenomics. So first of all, Symphyotrichum was traditionally treated as asters in the New World and later segregated into its own genus, Symphyotrichum. And then it is divided into five subgenera, and that has more than 100 species of naturally occurring hybrids of annual and perennial plants. So these are distributed across the Americas, some Caribbean, and one species in Eurasia, and is introduced worldwide. And this is well known for its complex taxonomic history, partly due to the high levels of morphological plasticity, suspected interspecific hybridization, and then polyploidy. It has been reported that more than 50% of these Symphyotrichums are polyploid in nature. Our earlier paper, where we did the phylogenomics investigation into biogeography of Mexico and Eastern U.S. disjunction in Symphyotrichum, we were able to delimit species in subgenus Virgulus. Here we recovered a well-supported phylogenetic tree where we included 23 out of 29 species, including 5 out of 6 Mexican species. So our sampling was nearly complete. And then here in this phylogenetic tree, we can see that Symphyotrichum defoliatum, a member of subgenus Astropolium, was recovered as sister to the subgenus Virgulus with high support value. And then the Western North American species, Symphyotrichum euchinense, was recovered as sister to the rest of the group. And the clade containing all the Mexican species was recovered sister to the clade containing some of the Eastern North American species with a high support value as well. And then the very similar relationship was recovered using the concatenated Raxamol tree, where we had more stronger support value than the astral tree. And then we went ahead and did the biogeographic analysis. So the most recent common ancestor of the subgenus Virgulus was reconstructed inhabiting the Western North America at around 1.60 million years ago. And then the several subclades diversified into different ranges. Most recently, at around 0.44 million years ago, the clade containing Mexican species diversified from the Eastern North American species. And now, after all that, my few things that I wanted to do was to revisit morphological delimitation and also use that morphometric data and the analysis of ecological niche shift in a phylogenetic context to test the two contrasting hypotheses. One, either the morphological and or niche conservatism, or the other one, totally different convergent biome adaptation. And furthermore, I wanted to analyze the correlation between habitat and morphology, and finally test whether lineages within subgenus Virgulus had undergone multiple independent transitions from one habitat type to another, indicating the role of habitat shift in adapting historical climate change. Okay, for that, our materials and methods included using a phylogenetic data. We used the same data that we recovered for our first paper using Angiosperm 353. And for the morphological data, we recorded the measurement for 13 morphometric characters for about 18 out of 29 known species. That is about 62% of species diversity. And we did this all using the herbarium specimens. And all our statistical analysis were done in R. We did the linear discriminant analysis to investigate and visualize the differences among the species in morphological characters. we did MANOVA to determine the overall group significance, and finally we did the principal component analysis. And for the ecological niche evolution, we downloaded the occurrence records from GBIF, and also we downloaded 36 environmental predictor variables that captured the aspects of climate, soil, land cover, and topography. For the habitat suitability predictions, we performed the species distribution modeling using Maxent, and we used those Maxent models to generate predicted niche occupancy profiles. And from that, we calculated the mean of all extracted occurrence values so that we could visualize niche evolution throughout the phylogeny using our package PhyTools. And to test for the phylogenetic signals, we use Pagel's lambda and Bloomberg's K statistics. And for the correlation analysis, we use Mantel test using the Spearman correlation. So that will give us the correlation between phylogenetics, morphometrics, and the environmental distances. And for the habitat classification, we did kind of a different approach where we converted quantitative niche descriptors from our environmental data into categorical habitat classification. For that, we constructed a matrix of pairwise Euclidean distances between species across all 36 predictor variables. And then we used that normalized version of this matrix to perform the k-means clustering that groups the species into k clusters and produces the categorical classification from 0 to k minus 1. And now for the habitat reconstruction, we classified that. Uh, sorry, was that a question or?
I think there's a little bit of feedback from one of the people here. Just carry on. Sorry.
Okay. Okay, then we use this classified habitat using results from k-means clustering and median data from environmental data to establish qualitative descriptors for the clusters. And then we finally use the R package biogeovirs to reconstruct the habitat shift evolution through time. So now into the results, our morphological variation from the linear discriminant analysis, so the significant or statistically significant variation with the overall classification success of about 79%, with a high MANOVA value for most of the variables. However, there were some exceptions. Here, this graph for the simplicity, I divided this graph into different sectional analysis where we can see the differences between the analyzed species. And now from our PCA analysis, we saw that the secondary axis leaf length and width, along with ray length, were the highly representative variable in describing this morphological variation in this subgenus. And similar to the morphological variation, the LDA showed the statistical significance in environmental variation with the overall classification success of 90% with a high MANOVA value. Same thing as the morphological variation. We divided this graph into sectional analysis to see more of the variation between species. And here the climatic, like such as the temperature and precipitations, were highly represented variables. And now for the phylogenetic signal test, all of our phylogenetic signal tests were insignificant for overall morphological data. That means it is suggesting that a stronger overall role of convergence than phylogenetic history. However, if we looked into individual characteristics, few of these, secondary axis leaf length and width, below capitulum hair length, those were highly conserved throughout time in phylogeny. And for the environmental variables, the phylogenetic signal was again insignificant, that suggested that the niche analyzed as a unit is not phylogenetically conserved. And again, the individual niche parameters, our 36 environmental parameters we measured, some of them, such as bio 8, 10, 18, all these were highly significant. So they were highly conserved throughout the phylogeny. For the correlation analysis, we performed the Mantel tests and we found out insignificant correlation again. So there was no correlation when we looked at environmental distance versus morphological distance or environmental differences versus the cophonetic distance or morphological distance versus cophonetic distances. So when we put this all together and analyzed as a partial Mantel test, we saw the insignificant statistical differences again. So what that suggests us was that overall morphological and environmental variation are largely independent of phylogeny and each other. So they are not dependent. So for the habitat classification and ancestral reconstruction, our classified habitat number 3 was most likely ancestral state, suggesting that the ancestral origin in moderate temperature and rainfall for subgenus Virgulus. And also from this biogeobears, the figure, we see that the multiple major habitat shifts through time, yet some species such as this Amethystinum concolor, fanlary, they maintained the ancestral habitat despite the climatic volatility of the Pleistocene period in which they radiated. And now, finally, we wanted to see the correlation between habitat type that we classified and the morphological analysis. So the LDA recovered the overall classification success of about 33% for the morphological data and classified habitat types with significant MANOVA value. So this suggests that the morphology is habitat dependent. So the different species in similar habitat can have similar morphology in subgenus Virgulus. And now for the take-home message, our insignificant phylogenetic signals, with some exceptions, of course, suggested the independent evolution of most of the examined traits and variables and a complex and possibly discordant relationship of morphological aspects with environment and evolutionary history. And we also found the evidence for morphological evolution shaped by a habitat rather than phylogeny in our group. So we found evidence for multiple major habitat shifts through time, yet some species maintained the ancestral habitat despite the climatic volatility of the Pleistocene period, as shown by our previous biogeographic study paper as well. So now the next will be to further study focusing on the role of biotic interactions in understanding the drivers of evolution in Symphyotrichum to complement the abiotic framework presented here. All right. By that, I would like to thank you all for listening, and then I'm ready for questions.
Thank you so much. What a nice talk. And it's cool to see a lot of your PhD work come together. Are there any questions at the time? Feel free to drop them in the chat or unmute yourself. Are there any questions for either of the speakers as we wrap up? No, those were two really excellent talks. And yeah, as we close, I just want to really thank everyone for joining our last TICA Talk of the year. And by giving a shout out to the 2025 TICA Talks Committee that includes Abby, Isaac, Colin, Carol, and Erica. Erica was instrumental in organizing the organizers most of the time. So with that, I just want to thank everyone again, and I hope you have a nice holiday season. And I hope to see a lot of y'all at the TICA events in 2026. I think we can stop recording.